All right. So thank you all very much for coming out. So I'm Frank Nothaft. I'm a third-year PhD student in the AMP Lab, and I work at the intersection of computer science and genomics. So I'll be talking to you today about ADAM. So ADAM's a system that we've been building for doing large-scale parallel genomic analysis. So everybody nowadays is talking about genomics and precision medicine, and I think many of us know at some sort of abstract level what your genome is. And the simple way to think about it is that genomics is, your genome is the source code for life. It's about 3.2 billion letters long, and it's split across 46 files, 23 that you get from your mother, 23 that you get from your father. So these files in biology we call chromosomes, but for here we can abstract it away as just a bunch of long character sequences. One of the things that's very interesting about genomics, what makes it very different from programs, is that every single copy of this program for life is actually largely the same. Whether it's mine, yours, we're all about 99.9% the same. On average, we differ at one of every 1,000 sites. Additionally, most of the source code is comments. The actual part of the genome that codes for proteins is less than 0.5% of this whole 3.2 billion letters. The big questions that we're looking at, though, when we have your genome, we go ahead, we analyze it, because we're looking for these 1 in 1,000 position differences to see how they associate with the different traits and the diseases that we're affected with. We're not just looking for differences that happen in the part of the genome that code for a protein. This area, this 99% of the genome that doesn't actually code for proteins, it's not just comments. We know that it does have an effect on traits, even though it's not a direct effect. So the way that this works, I assume that most of you aren't familiar with sequencing machines, is it's actually a simple thing to reason about. It's a Poisson sampling process. So when we go ahead, we sequence your genome, what we do is we divide this 3.2 billion letter source code into a bunch of short fragments. We duplicate them a bunch of times, and then we essentially pick 100 base long fragments out of that. So we're going to go ahead and reconstruct this 3.2 billion letter long book out of 100 base long sequences. And if we want to get 30 copies at each position in the genome, we can do that for about $1,000. So that was a very big accomplishment. It's actually, since the Human Genome Project occurred in 2001, the cost of sequencing a human genome has dropped by over 10,000 times. And so that's the progress that we've seen in 15 years. And we expect to see costs dropping, not necessarily as fast as they've been in the past 15 years. We're not going to see another 10,000x, but we'll see sequencing costs continue to improve. And the big implication here is that while in 2001, when we had this Human Genome Project come up, we looked at 20 individuals' genomes, and this cost about $1 billion to do. Now that we have this very high quality understanding of what the human genome is, we can use a simpler process. So what the Human Genome Project did was it generated this consensus for what every single one of our genomes look like on average. As we said earlier, 999 out of 1,000 places, we match more or less. From this, what we do is we'll take your individual genome sequence, we'll go ahead, compare it to what we call the genome reference, and we'll compute a diff. So this leaves us with two big problems. And the first is, how do we go ahead and compute the diff? And then the second is, how do we go ahead and make sense of the differences? So while this is much cheaper from a cost perspective, it's still fairly expensive to do. What we're doing is we're actually going ahead and we're going to take every single one of these sequences. We're going to align it. You can think of the text on the top, the reference copy. We're going to align every single fragment to this reference copy. And then we're going to run a statistical algorithm that goes ahead and finds a consensus sequence at every single place. So in this slide, I don't have any errors in the text. When you look up here, it's all good words. I didn't misspell worst when I copied it. When we look at a real human genome, though, in addition to the differences that we expect to see, this 1 in every 1,000 positions, we actually have an error rate of 2%. So we have errors in our sequence that are more common than the variants that we expect to see. So that's kind of what we're trying to go ahead and factor out. This whole process is actually fairly expensive. So what we went ahead and did in ADAM was we came up with a greenfield approach to this problem. So this right here is a picture of the reference genome. It's the best practices genome sequencing pipeline. So this is called the Genome Analysis Toolkit. It comes out of the Broad Institute of Harvard at MIT, and it's one of the most widely used tools in genomics. So when you're going through this process, we have this first stage up here that we're going ahead, we're doing the sequencing, this Poisson subsampling process, $1,000 gets us our data. The next steps from right here where we hit this map to reference, this is the string matching problem, all the way to the end where we actually get this diff, takes about 150 hours to run and can be about $500 worth of compute. So it's a very expensive process, and people have really optimized this for this case where you have a single machine that's very big that you're going to store all your data on, you're going to process it on a single machine. You're not going to go very big, you're not going to go parallel. Unfortunately, this has led to several very big problems in the genomics community. Since people are building in this environment, they're traditionally building for things that look like HPC-style compute clusters. This process is actually I/O bound. When we talked about generating the 30x replica of the human genome, that consumes about 100 gigabytes of data. So we're going to go ahead, we're going to read 100 gigabytes of data to do this compute process, and right now, every single time we run a stage, we write it to disk. So we're violating a lot of these assumptions that we preach for in Spark. And if we're building in an HPC environment, if we did want to go parallel, we're going to wind up being constrained by the storage bandwidth that we get going to a shared file system. A lot of these pipelines also make interesting assumptions about how data is laid out. There are a lot of optimizations, both in the file formats that people use and the way that we traverse over our data that are really specific to this one application. They do very interesting optimizations by pushing down sort order invariance into their data, and this winds up making it very hard to go ahead and combine tools. So while I could run the GATK, let's say I wanted to sub out one of these stages. Someone comes up with a new method, I have to go ahead and figure out, is the sort order they're using in their method compatible with the sort order I'm using in my method? It makes it very hard to combine tools. It makes it very hard to run tools in parallel. So we went ahead and we took a step back, and we did what many people who are educated in simple database design did, and we decided to factor everything out and just come up with a schema that defined the genomic data that we were working on. This is actually the first thing that we did in Atom, and it's been very interesting because some parts of genomics have very well-defined schemas, some parts do not. So I'll talk a little bit more about that later. From that, the schema provided a very nice narrow waist that we could work against. So if I'm writing some genomic algorithm, I'm going to have an RDD, and that RDD is going to be full of of just little objects that have this schema, right? We actually use Avro for our serialization, but you can load up our data in a data frame, doesn't matter. On top of this, we've gone ahead and we've accelerated a lot of the common access patterns with higher level primitives. So one of the things that's very common in genomics is you do this very special type of a join. So the thing that's interesting about your genome is that as I said earlier, you have 23 chromosomes. You get 2 copies of each. One from your mother, one from your father, and a lot of things in genomics are described by where they are on a specific chromosome. So, you might be interested in a gene that is on chromosome 17, position 235,000, right? So very, very frequently, we're going ahead and we're taking, genomics data set 1. Maybe it's a set of areas where I am different from the average human. And genomics data set 2, it might be a description of all of the genes that are in the human genome. And we're doing these joins on them. We're doing them fast. We want to do them fast, but it's not necessarily something we can do fast natively. The naive implementation in Spark would actually take about 90 hours to run, on a fairly reasonably sized cluster. But the optimizations that we've made takes that 90-hour runtime, brings it down to 1 hour. And there are other nice things about this. This is a very common pattern that people use in genomics. But as I said earlier, the way they accelerate it is by pushing down these sort order invariants into their file formats and writing against these very low-level abstractions. So when we're going ahead and building Atom, we verified performance by rebuilding some of these canonical algorithms that people are using to run, take 150 hours to run. When we rebuilt the algorithms, we found that not only were we able to come up with good ways to scale them in parallel, but we actually identified several bugs in the algorithms that came because they were writing at this low level against this sorted iterator across the genome. By writing at a higher level, it made it, one, much easier to write the code, and two, eliminated bugs in about 4 or 5 of the stages that we were interested in. Additionally, we've gone ahead and we've, after designing our schemas, we decided to pick an appropriate storage format for the data. As I've discussed, a lot of the storage formats in genomics are optimized for the traditional use case where you have one read, you have 2 reads, you essentially have a row-oriented storage. However, this isn't necessarily a great fit for science. We're frequently going ahead and slicing and dicing our data set. It's actually very common that the first analysis that you run is only going to look at about 6 fields out of the 33 field schema that I was showing a couple slides back. The 6 fields that we're looking at here are less than 2% of the data stored on disk. By going to a columnar storage format that allows us to go ahead and project fields out as we need them, we were able to get just a 10x performance increase right off the bat, and we were able to improve how our algorithm actually scaled out. Additionally, we're frequently running selective predicates. So while I might be interested in analyzing the whole genome, someone else might come along and say, oh, I know I'm only interested in chromosome 1. If they're only interested in chromosome 1, they can go ahead and throw away more than 90% of the data just by applying that one filter function. And that's something we can very easily optimize by using columnar storage. One of the things that's been very interesting as we've gone through the Atom project is that many people have criticized our choice of going ahead and rewriting our schemas and using a different columnar storage format on disk instead of these legacy file formats. Actually, we've several times had people tweet this exact figure at us. And the thing that's actually really interesting, and this is a really subtle point about the stack design that we use, is that by putting the schema as the narrow waist, it's easy for us to go ahead and write our algorithms against our optimized storage format. Well, it's not optimized, it's just a commodity columnar store. We use Parquet. But it's very easy for us to go ahead and run our algorithms on any of these legacy data formats. So in genomics, there are about 5 different key types of data. Some of them have 1 or 2 well-accepted formats, but in other of these sets of data, you'll have more than 5 or 6 different formats that describe the same basic model. So by having this, having the schema in the middle here and being able to switch out how our data is materialized on disk, we actually are able to work. We essentially provide anybody who's writing an algorithm that processes genomics data in Atom with, they can be agnostic as to how their data is actually stored on disk. They can save it into our legacy, into our columnar format. They can save it to any of the legacy formats. They can read from any of the legacy formats. So it gives them a lot of flexibility. They don't have to worry about whether their algorithm interoperates with something by reading the same bytes off of disk. So all of this architectural stuff is nice, and it provides great benefits if you're going ahead and writing these genomic algorithms. And you don't want to worry about writing at a very low level or worry about what your exact bytes look like on disk. But nobody really cares if it's not fast, right? So again, our big mission at the start was to take this 150-hour pipeline and to get it to be not 150 hours. So we focused on, on these stages that are here in the middle because they're very expensive stages. In the 150 hours, you actually spend about 100 to 120 hours just in these 3 stages. So, this is kind of a performance result that we had from a paper that we presented at SIGMOD. We achieve linear scalability on a single genome out to 128 nodes. So here we're evaluating ourselves on a 250 gigabyte data file. So this is one of the largest human genomes and it's a human genome that people use to evaluate many tools on. So it's a kind of a standard data set that's used. On a single node, what we did was we compared our performance on a single big node to the state-of-the-art tools on the single node. We ran several of these different stages. We picked the fastest performance of any other tool out there, and we compared against our performance. On a single node, we would be up to 3x faster, and then we would scale out linearly to 128 nodes. And on 128 nodes, keep in mind this is about 200 megabytes of data per machine. So we're operating with a very small amount of data per machine, and we're still getting a very good parallel speed out. Since we've gone ahead and run these experiments, we've actually made a lot of single-threaded performance improvements inside of Atom that have given us between a 1.5 and 2.5x improvement uniformly across our code. So that would mean, we're no longer 3x faster on a single node, but we're about 6 to 9x faster. One of the great things about working on this project is that we've had many collaborators that are out there. So Atom is a project that has about 46 collaborators in the open source field. Many people here at Berkeley, but a few folks at other hospitals such as Mount Sinai in New York, and people in industry at Cletter and Microsoft Research. If you're interested in genomics, or you'd like to talk more about how you can scale genomic algorithms out faster, please feel free to find me later, and I would love to talk and let you know about how you could contribute or use our code. Any questions?